Welcome, medical students and everyone joining us for the deep dive. If you're listening, you're ready to really get into the core concepts of medical parasitology. We're talking about those fundamental ideas that really explain how these organisms survive, how they spread, and ultimately how they make us sick.
Absolutely. This is foundational stuff. And this deep dive draws heavily on the excellent notes from Dr. Mohammed Wiesman. He's a medical lecturer at the Medical Parasitology and Entomology Unit, Faculty of Medicine and Defense Health at the National Defense University of Malaysia, UPNM, in Kuala Lumpur.
So our goal today is pretty clear. Map out that constant back and forth, that evolutionary arms race between hosts and parasites. We need to define who track their life cycles and really understand the two big outcomes: disease or pathogenicity, and how our bodies fight back, immunity.
Right. And we've got 7 key learning outcomes we're aiming for today, basically. Defining terms, differentiating parasites, understanding vectors, the host-parasite relationship itself, life cycles, transmission and disease, and finally, the clinical approach. It sounds like a lot, but it all connects.
Okay, let's start with the basics then. The parasite itself, we know it needs things from us: food, shelter, reproduction. But how important are those subtle differences, like whether they live on us versus in us?
Oh, that's absolutely critical. Where they live, that location dictates pretty much everything: how they cause disease, how they transmit, and importantly for you, how we control them. So we broadly group them into 3. First, you've got ectoparasites. Think external. They live on the surface: skin, hair, lice, ticks, mites, those kinds of things. Transmission is often direct contact or sometimes vectors.
Okay, surface dwellers. Makes sense.
Then you have endoparasites. These are the internal ones. They get inside blood, tissues, organs. Think malaria parasites in red blood cells or tapeworms in the gut. Their transmission is way more complex. It could be ingestion, vector-borne like mosquitoes, or even direct penetration through the skin.
And then there's that third group, the one that can be a bit tricky for diagnosis: the free-living parasites.
Right, the outliers. These guys normally live perfectly happily out in the environment: soil, water, not bothering anyone. They're not typically parasitic. The key clinical point is they become parasitic opportunistically. Maybe the host's defenses are down, or they accidentally get into a place they shouldn't, like the eye or a wound.
Like acanthamoeba.
Exactly. Acanthamoeba causing keratitis is the classic example. It's usually free-living, but get it in a contact lens wearer's eye. Big problem.
Okay, that location difference is clear. Now let's dig into dependency. This feels important for eradication strategies. What's the real difference between an obligate parasite and a facultative one?
It really comes down to survival dependency. An obligate parasite, think Plasmodium formularia, absolutely must spend at least part of its life cycle inside a host. It cannot complete its life or reproduce without one. They're completely reliant on that host chain. So control often means breaking that chain.
Makes sense. No host, no parasite?
Pretty much. But a facultative parasite, like acanthamoeba again, or maybe Naegleria fowleri, they can live as a parasite, but they don't have to. They can survive and reproduce quite happily as free-living organisms in the environment.
So control is harder. You have to worry about the environment too, not just infected people.
Precisely. Environmental decontamination becomes just as important as treating the patient for facultative parasites.
Okay, and sometimes parasites get lost. Can you illustrate the difference between an accidental parasite wrong host and an aberrant parasite wrong place?
Yep.
Using, say, Echinococcus granulosus and Toxocara canis?
Yeah, this is a really important distinction for understanding the resulting disease. An accidental parasite is one that infects a host it doesn't normally infect. It's the wrong species for it. Humans getting infected with Echinococcus granulosus, the dog tapeworm, is a prime example. The parasite gets into us, usually via eggs from dog feces, but it can't complete its life cycle. It gets frustrated, essentially, and often forms large cysts, like hydatid cysts, causing severe disease because it's in an environment it wasn't designed for.
So, wrong host leads to abnormal development and disease. What about aberrant?
Aberrant means the parasite might be in its normal host or even an accidental one, but it wanders off track. It migrates to the wrong tissue or organ within that host. So Toxocara canis, the dog roundworm, when it accidentally infects humans again, usually kids eating contaminated soil, the larvae don't mature. Instead, they migrate. They can end up in the liver, lungs, brain, or eye, causing visceral larva migrans or ocular larva migrans. The damage comes from this abnormal migration, this wandering into the wrong location.
Right, so Accidental is wrong host altogether. Aberrant is wrong location within a host.
Got it. Okay, let's flip the coin. We've defined the parasite, now let's define the host. The parasite needs hosts to complete its life cycle, right? So we categorize hosts based on what the parasite needs to do there. Let's start with the 4 main developmental roles: definitive, intermediate, paratenic, and transport.
Exactly. It's all about the parasite's life stages, particularly reproduction. The definitive host is, well, the definitive one. It's where the parasite reaches sexual maturity and reproduces. For Plasmodium, the malaria parasite, the definitive host is actually the female Anopheles mosquito, because that's where the sexual cycle happens.
No, right, not us.
Not us. We're the intermediate host for malaria.
Right.
This is a host where the parasite undergoes some necessary development, like asexual replication, but it doesn't reach sexual maturity.
Okay, so definitive for sex, intermediate for development. What about paratenic and transport? They sound a bit like helpers. They kind of are. Helpers for survival and transmission. A paratenic host, sometimes called a transport host too, but there's a nuance. It's a host where the parasite doesn't undergo any further development. It just hangs out, waiting. Think of it as a temporary parking spot. But crucially, this host isn't essential for the life cycle to complete, but it can help bridge the gap to the next required host, often increasing the chances of transmission.
Like rodents carrying toxicar larvae. The dog eats the rodent, gets the worm.
Exactly. The larva doesn't develop in the rodent, but the rodent helps get it to the dog. Now, a pure transport host is even simpler. It's just a mechanical carrier. The parasite undergoes 0 development. It's literally just hitching a ride. A classic example is a cockroach or fly carrying Entamoeba histolytica cysts from feces onto food. The fly isn't infected, it's just contaminated.
Like a dirty taxi.
Precisely, a disease taxi.
Okay, that covers the hosts involved in the cycle itself.
Yeah.
But for public health and tracking diseases, there are other host types we need to know: reservoir, accidental, and dead end. How do these fit into the picture?
These terms are crucial for epidemiology, understanding where the infection persists and how it spreads to humans. A reservoir host is key. This is an animal, or sometimes human, that harbors the pathogen, often long-term, usually without getting sick itself. They act as a continuous source of infection for others, including vectors or accidental hosts like us. Think about monkeys and Plasmodium knowlesi malaria in Southeast Asia. The monkeys are the reservoir.
Keeping the infection going in nature.
Exactly. Now, we already touched on accidental host. That's just a host, often human, that gets infected but isn't part of the parasite's usual life cycle, like us with Echinococcus or Toxocara.
And a dead end host. Sounds final.
It usually is, for the parasite at least. A dead-end host is one where the parasite can infect, maybe even cause disease, but it cannot be transmitted onwards to another host, including the definitive host. The life cycle stops there.
So humans with Echinococcus are both accidental and dead end hosts.
Often, yes. We get infected accidentally, and the parasite can't develop properly or transmit out of us to complete its cycle in dogs. The infection terminates with us. That's why it's called a dead end.
Right, that clarifies the host landscape. Now let's talk about how parasites move between these hosts. Vectors are obviously huge here. What's the absolute key difference medical students need to remember between a biological vector and a mechanical vector?
This is fundamental for understanding transmission. A biological vector is not just a taxi. It's an essential part of the parasite's life cycle. The parasite must undergo some development or multiplication inside the vector before it can be transmitted. The mosquito-carrying malaria is the perfect example. Plasmodium has to develop inside the mosquito before it becomes infectious to humans. The mosquito is effectively another host.
So the vector is actively involved.
Correct. Whereas a mechanical vector is purely a physical carrier. It picks up the infectious agents, say bacteria or parasite eggs on its feet or mouth parts, and passively transfers it to a host or food. There's no biological development of the parasite within the mechanical vector. That fly landing on feces and then your lunch carrying Shigella or maybe helminth eggs, that's a mechanical vector.
Gross, but clear. Okay, stepping back a bit. Parasitism is just one type of long-term interaction between species under the umbrella of symbiosis. Can you quickly sketch out that spectrum from mutualism to commensalism to parasitism?
Sure. Symbiosis just means living together. It covers a range of relationships based on who benefits and who, if anyone, is harmed. At one end, you have mutualism, where both partners benefit. Think bees pollinating flowers. Bees get nectar, flowers get pollinated, win-win.
Okay.
Then there's commensalism. Here, one organism benefits and the other is essentially unaffected, neither benefits nor is harmed. The classic example is remoras attaching to sharks. The remora gets scraps and transport, the shark likely doesn't even notice, neutral for the shark. And finally, parasitism, which is our focus. One organism, the parasite, benefits, gets food, shelter, strictly at the expense of the other organism, the host, which is harmed in some way.
Clear scale. Both win. One wins in neutral, one wins and loses.
Exactly.
Okay, let's connect this back to human health with the One Health concept. We hear this term a lot now, especially with zoonotic diseases, infections jumping from animals to humans. What does One Health mean when we're dealing with parasites like Toxoplasma from cats or Nalissi malaria from monkeys?
It's a really important shift in perspective. First, let's define zoonosis, an infection naturally transmissible from vertebrate animals to humans. And related is opportunism, where parasites, sometimes zoonotic ones, take advantage of a weakened host, like Cryptosporidium causing severe disease in AIDS patients. The One Health approach recognizes that human health, animal health, and environmental health are inextricably linked. For zoonotic parasites like Toxoplasma or Nalissi malaria, you can't just focus on treating infected humans.
You have to look at the source.
Precisely. You have to understand the parasite's life cycle in the animal reservoir. Cats for toxo, monkeys for Nalissi. You need to consider the ecology. How do humans come into contact with the animals or contaminated environments? What are the agricultural practices, deforestation patterns, animal behaviors involved?
It's the whole ecosystem.
Exactly. One Health demands collaboration between physicians, veterinarians, ecologists, public health officials, everyone involved in that human-animal environment interface to effectively control the disease. It's holistic.
Okay, that makes perfect sense. So we've got the players, the relationships, the transmission routes. Let's talk about the parasite's journey, its life cycle. How does a simple monoxenic or direct life cycle compare strategically to a more complicated polyxenic or indirect one? Let's use pinworm versus malaria as examples.
It's basically a difference in complexity and reliance. Monoxenic means the parasite needs only one host species to complete its entire life cycle. It's direct transmission, usually host to host. Enterobius fumicularis, the pinworm, is a great example. It lives in the human gut, lays eggs around the anus, these contaminant fingers or bedding, and are ingested by the same or another human. Simple, direct, only involves humans.
Very efficient, if unpleasant.
Highly efficient in crowded conditions, yes. Now, polyxenic or indirect means the parasite requires more than one different host species to complete its life cycle. Plasmodium malaria is the classic. It needs both a human intermediate host for asexual stages and a specific mosquito species, definitive host for sexual stages.
So it's more complex, more steps involved. Is that a disadvantage?
It can be. There are more points where the cycle can be broken. If you eliminate the mosquito vector, you stop malaria transmission. But the advantage for the parasite is often wider dispersal and the ability to exploit different niches or environments through its different hosts. It's a more complex strategy, but potentially more resilient in some ways.
Right, complexity versus simplicity. Now, the practical part, getting into the host. Transmission. There's so many ways. What are maybe three absolutely critical modes of transmission that medical students must have on their radar?
Okay, if I had to pick three high-yield ones. First, definitely fecal oral transmission. Super common. This is where infective stages like cysts or eggs passed in feces contaminate food, water, or hands and are then ingested. Think Entamoeba histolytica, Giardia lamblia cysts, or Ascaris lumbricoides eggs. Poor sanitation is the driver here.
Got it. Fecal oral. What's number two?
Second, skin penetration. Some parasites have larval stages that live in soil or water and can actively burrow through intact human skin. No ingestion needed.
Yeah.
Hookworms, Necator, Ancylostoma, Strongyloids, Stercoralis larvae in contaminated soil, or Cystosoma cercariae in fresh water. They just drill right in. Walking barefoot or swimming in infested areas is the risk.
Yikes. Okay, skin penetration. And a third major route often involves what we eat, right?
Absolutely. Third is ingestion of an intermediate host or tissues containing infective stages. This usually means eating raw or undercooked meat or fish that's harboring parasite larvae or cysts. Taeninia tapeworms from infected beef or pork, T. saginata, T. solium. Toxoplasma gondii from cysts in undercooked meat. Diphyllobothrium fish tapeworm from raw freshwater fish. Cooking food properly is key here.
Fecal oral, skin penetration, ingestion of infected tissue. That covers a lot. We should probably quickly mention autoinfection, too.
Yes, definitely worth a mention. Autoinfection is where a parasite can complete its entire life cycle within a single host, leading to reinfection without ever leaving the body, or by reinfecting immediately after eggs are laid externally. Strongyloids is the terrifying example, especially in immunocompromised people. The larvae can mature and repenetrate the gut wall or perianal skin, leading to a massive increase in worm burden hyperinfection, which can be fatal. Pinworm also has external autoinfection.
Okay, good to keep in mind. So sources are things like contaminated soil, water, undercooked food, and of course, blood-sucking insects acting as vectors.
Those are the big categories, yes.
Right, the parasite is in, now the damage begins. Pathogenicity, the ability to cause disease. What are the main ways parasites actually harm the host? Is it direct physical damage, resource theft, or is it often our own immune system causing problems?
It's often a combination, but we can break down the mechanisms. First, simple mechanical damage. This is physical obstruction or trauma caused by the parasite itself, a large mass of Ascaris worms blocking the intestine or bile duct. Schistosoma eggs getting lodged in liver blood vessels, blocking flow and causing portal hypertension. The physical presence causes problems.
Just being there is harmful.
Exactly. Second, nutrient competition. The parasite basically steals the host's food. Giardia interferes with fat absorption in the gut, leading to malabsorption and fatty stools. The fish tapeworm, Diphyllobothrium laudum, has a huge appetite for vitamin B12, leading to megaloblastic anemia in the host.
Stealing vital resources.
Yep. Third mechanism, tissue invasion and destruction. Some parasites actively invade host tissues, causing direct damage, inflammation, and necrosis. Entamoeba histolytica invading the colon wall, causing ulcers, or migrating to the liver and causing a liver abscess. That's direct destruction.
Okay, mechanical, nutrient theft, direct invasion. What about the immune system's role?
Ah, yes. Immune-mediated damage. This is crucial. Often the most severe pathology isn't caused directly by the parasite, but by the host's own inflammatory and immune response to the parasite or its products. The granulomas that form around schistosoma eggs in the liver or bladder wall are a prime example. The immune system is trying to wall off the foreign eggs, but the resulting fibrosis and inflammation cause the organ damage and disease symptoms.
So our own defense causes collateral damage.
Significant collateral damage, frequently. We should also briefly mention toxin production. Plasmodium releasing toxins during red cell rupture contributes to fever and shock in malaria. And rarely, but importantly, tumorigenesis, chronic inflammation from certain parasites like Schistosoma amatobium in the bladder can increase the risk of cancer over time.
Wow, a whole arsenal of ways to cause harm. Now let's look at our defense. Immunity. We often hear about Th1 versus Th2 responses. What's the fundamental difference, and which response targets which type of parasite generally?
This is a core concept in immunoparasitology. It's about tailoring the immune response to the type of threat. The Th1 response is primarily cell-mediated immunity. It involves T helper 1 cells releasing cytokines like interferon gamma, IFNDO, which activate macrophages and cytotoxic T cells. Its main job is to find and destroy host cells that are infected with the intracellular pathogens parasites living inside our cells. Think Leishmania, Toxoplasma gondii, Trypanosoma cruzi. The T1 aims to kill the infected host cell to eliminate the hidden parasite.
Okay, Th1 for intracellular threats. What about Th2?
The Th2 response drives humoral immunity and responses against larger extracellular pathogens. T helper 2 cells release cytokines like IL-4, IL-5, and IL-13. These promote B-cell antibody production, especially IgE, and crucially, they recruit and activate eosinophils and mast cells. This response is geared towards tackling things outside cells, particularly large extracellular helminths, worms like Ascaris, Schistosoma, hookworms. IgE coats the worm, eosinophils bind to the IgE and release toxic granules to try and damage or expel the large parasite.
So Th1 is intracellular kill, Th2 kills extracellular expulsion or damage, especially for worms.
That's a good way to summarize the main thrust, yes.
But these strong immune responses can backfire, leading to hypersensitivity reactions. Can you give a quick example for each of the 4 types of hypersensitivity we might see in parasitic diseases?
Sure. These are basically exaggerated or inappropriate immune responses causing tissue damage. Type 1, Ig-mediated allergic. Think immediate reactions. Swimmers itch the rash caused by skin penetration by avian Schistosome cercariae, or potentially life-threatening anaphylaxis if a hydatid cyst, Echinococcus ruptures, releasing massive amounts of antigen.
Okay, type 1 is allergy. Type 2.
Type 2, antibody-mediated cytotoxicity. Antibodies bind to host cells, leading to their destruction. A key example is the anemia in malaria, where antibodies contribute to the destruction of both infected and uninfected red blood cells.
Right, type 3.
Type 3, immune complex-mediated. Antigen-antibody complexes form, circulate, and deposit in tissues, causing inflammation. The classic example is kidney damage, like nephrotic syndrome seen in chronic Plasmodium malaria infections due to immune complex deposition in the glomeruli.
And finally, type 4.
Type 4, cell-mediated delayed type. This is T-cell driven, takes longer to develop. Granuloma formation around cystosoma eggs is a type 4 reaction. Also, the chronic inflammation and fibrosis leading to conditions like elephantiasis in lymphatic filariasis involves type 4 mechanisms.
So all 4 types of hypersensitivity can be triggered by parasites. It just shows how complex the interaction is.
Absolutely.
Given how sophisticated our immune system is, how do parasites manage to survive? They must have evolved some pretty clever evasion tactics. Can you give us 3 key ways they hide or fight back?
Oh, they're masters of evasion. It's an ongoing evolutionary arms race. 3 major strategies. First, intracellular location. Simply hiding inside host cells puts them out of reach of many immune components, like antibodies and complements. Plasmodium invading liver cells and then red blood cells is a prime example. Leishmania living inside macrophages, the very cells meant to kill them.
Hiding in plain sight, inside our own cells. Clever.
Second, antigenic variation. This is like changing disguise constantly. Some parasites, most famously the African trypanosomes, Trypanosoma brucae, that cause sleeping sickness, continuously switch their major surface coat proteins. Just as the host mounts an antibody response to one coat, the parasite switches to a new one, rendering the previous antibodies useless. This leads to waves of parasitemia and fever. Plasmodium falciparum also does this, to some extent, with its surface antigens on red blood cells.
Always one step ahead. It's a third tactic.
Third, antigenic mimicry or disguise. Some parasites coat themselves with host molecules, essentially camouflaging themselves as self to avoid immune recognition. Schistosoma worms are known to acquire host blood group antigens and MHC molecules onto their surface. It makes it harder for the immune system to recognize them as foreign. Other strategies include shedding their antigens, suppressing immune responses directly, or forming protective cysts.
Incredible evasion strategies. Okay, let's bring all this knowledge back to the clinic. A patient presents. You suspect a parasitic infection. How should a medical student or clinician approach this? What are the essential steps?
Right. The clinical approach needs to be systematic, especially because symptoms can be vague or mimic other diseases. There are essentially 4 key steps. 1, history taking. This is absolutely crucial and often gives you the biggest clues. You need meticulous detail. Where have they traveled? When? What do they do there? Occupation? Hobbies? What's their diet, like raw fish, undercooked meat? Any exposure to fresh water? Animal contact? Symptoms might be subtle or nonspecific, so a thorough history is paramount.
Don't skimp on the history. Got it. 2.
2, physical examination. A general exam, of course, but also focus on systems potentially involved. Look for rashes, lymph node swelling, head and neck. Check the abdomen for organomegaly, liver, spleen. Listen to the lungs. Check neurological status if relevant. Tailor it based on the history.
Okay, history, physical. What's next?
3, investigation. This is where the lab and imaging come in. Basic blood work first check for eosinophilia, a common clue for Helminths. Anemia. Then specific parasitology tests. Stool examination for ova and parasites. Multiple samples often needed. Blood smears for malaria or filaria. Serological tests for antibodies or antigens, maybe PCR. Radiology like ultrasound, CT, or MRI can be vital for finding cysts, like hydatid, or abscesses and amoeba. Sometimes endoscopy or even surgical biopsy might be needed for diagnosis.
So, targeted investigations based on suspicion. And finally,
4, treatment and management. Once diagnosed, this involves specific antiparasitic drugs, pharmacological, but don't forget non-pharmacological aspects. Supportive care, managing complications like fluid balance, nutrition, preventing further spread, and potentially treating contacts or addressing the source of infection, like water purification or vector control advice.
history, physical, investigation, treatment management. A solid framework.
Indeed. It requires putting all the pieces together, the parasite's biology, the host's response, and the patient's specific situation. So, to wrap up, this deep dive really gave us the essential framework for understanding these complex host-parasite relationships. We looked at differentiating the roles of both parasites and hosts and outlined those key mechanisms of disease, immunity, and how we approach it clinically.
That was incredibly thorough. A fantastic tour through the fundamentals. Thank you so much to Dr. Mohammed Wiesman for providing the detailed notes that made this possible. And thank you all for joining us on this deep dive. If you found this helpful, please do like, comment, share, and subscribe. Keep learning parasitology the fun way, and we hope you'll join us again for our next session. Bye for now.